Jai Hind, welcome to Bioaffairs. Today I'll be talking about virology. So I'm starting this. I have given some classes about replication and all, but I've decided to start a series on virology, and I will cover the important topics and important information regarding to viruses. So let's start. So here I'll be talking about what are viruses, their structure, morphology, nature, and some important examples. So first we have to know what is a virus. Virus is a microorganism which cannot be seen with unaided eyes. You need a microscope to see the virus. Most of the viruses can be seen only under the electron microscope, but some viruses like Mimi virus and Pox virus you can see under the light microscope with some modifications. Now viruses are, unlike bacteria, are acellular, means they do not have a cellular structure. So with a cellular structure, you know that in case of bacteria there are cytoplasms, then nucleoid structure, okay, there is cell wall, then cell membrane. So this is a kind of typical cell structure. If you consider eukaryotic cells, there are organelles and other things, but in case of bacteria, a virus, there is no such thing. So that is why virus is a acellular structure. Now virus is extracellularly and intracellularly, both in both way it can present itself. Extracellularly we do not know generally, but when it infects us, it gets multiplied itself and cause the disease, the pathogenic forms, then we'll know that we are infected with this kind of viruses. So these viruses reproduce within the living cells. It may be an animal cell, it may be a plant cell, it may be a fungi, it may be a bacteria. Okay, so we need to culture them to identify, and there are several other molecular techniques are there. But when you are talking about virus, a particular virus particle can be called as a virion. Remember that. Okay, so virus is an obligate parasite. That is also an important thing. Now the pathogenicity ranges, as I have told you, it can it not only infect humans or plants, but it infect bacteria also, and fungi, and some archaeal viruses can cause the disease to archaeobacteria. Okay, so this is a very wide range of infection capability of a single virus or the viral domain. Now, coming to the structure of virus, virus generally contain a nucleocapsid structure. Nucleocapsid, the name defines here that it will contain a DNA or RNA. So a virus can be a DNA virus or a RNA virus. Now, if you go in deeper, it can be a single-stranded DNA or RNA virus, or it can be a double-stranded DNA or RNA virus, right? So this comes to nucleo. Now coming to the capsid part. So capsid is a protein cell that covers the, or that protects the nuclear genome. That may be a DNA or the RNA. This is called a capsid. So nucleocapsid term is used for the viral particle as it shows the particular morphology. Now some of the viruses, apart from this capsid and nucleic acid, may contain an envelope, right? Generally animal and plant viruses contain this envelope. So this envelope is reside outside the viral structures. I will show you the picture in the next slide, so you will be seeing. So these non-enveloped viruses contain only the capsid and the nucleic acid part. But in case of the enveloped viruses, apart from these two, there is a lipid bimembrane or lipid membrane where several proteins can present in case of enveloped viruses. I'll show you the picture. Now, some of the viruses or most of the viruses you can see, as I told you, they do not contain any organelle like ribosomes and other mitochondria like that things. But some viruses may contain special enzymes, and they do not contain any kind of ATP generating mechanisms. So these things are very important. No cytoplasm, no other cellular processes can be done here. Now coming to the viral structure, first I will show you some of the structure. Let's see this. So here you'll see that this is a non-enveloped virus, right? There is no such structure outside the capsid. This is the capsid. This is the purple one is the capsid structure. This red one is the nucleic acid. It may be a DNA or RNA, right? So here double stranded. I suppose this is a DNA. Now this is called a non-enveloped virus, right? Then coming to the enveloped virus. Enveloped virus, I told you that outside this capsid and nucleic acid there will be an extra layer of lipid membrane. Now this lipid membrane may contain spike proteins. So spike proteins have several functions. I will talk about it in the later part. Nucleic acid, capsid and spike proteins. Apart from the spike proteins, there are other matrix proteins may present in this kind of envelope. But not all virus contains spike. Not all virus contains the spike proteins. Matrix proteins. So the composition of this envelope differs from virus to virus in the enveloped virus section. Right. Now let's see this picture. Here I have shown you several viral structures: vaccinia virus, herpes virus, rhabdovirus. So the morphology of the viral structure, you can see there is a difference, a huge difference among them, right? You see this is not a capsid structure, vaccinia virus. In case of herpes, this is a capsid. Outside that there is a membrane and these spike proteins are present. In case of rhabdo, it is a bullet shaped and the spike proteins are present. In case of this is a bacteriophage, so it contains icosahedron capsid as well as this slender hollow tube, then tail pins or base plates, like that the complex structure is there. Now this is also bacteriophage. Bacteriophage means those viruses that infect the bacteria are bacteriophages, right? Now this is a spindle shaped viral structure. Now again only nucleocapsid structure. This is also nucleic acid structure, and this is a tobacco mosaic virus, RNA virus. It is a hollow cylindrical structure. Within that the nucleic acid is present. This is the Mimi virus, very large virus, you can see under the light microscope, and this is a typical two-tailed virus. So you need to remember these names for your MCQ questions. Okay, so the variety in their cellular structure, acellular structure or the capsid structure, you can see here. Now let's coming to the particular capsid variety. So virus, as you can see, several capsids are there. There is structural, you can say the structural variety of a viral family. Now mostly three types of structure are present, that is helical capsids, icosahedral capsids and capsids with complex symmetry. Complex symmetry means the capsid which do not encompass either helical or icosahedral completely. So in case of helical capsid, the hollow tube is present. I have shown you the picture of tobacco mosaic virus, and within that the genome is present. In case of icosahedral, remember that you can pause and go back. This icosahedral structure, the enveloped or non-enveloped viruses containing this structure. Now these triangular faces which I have shown you, the triangular faces, there are 20 equilateral triangular faces and 12 vertices. So if you consider this, the corner angular regions are called vertices, okay, and others are, and the 20 equilateral triangular faces, this region faces, okay. Now this capsid structure, if you see in the molecular level, the lowest formation is 5 or 6 protomers which contain the capsomers. So first is capsomer, which is composed of 5 or 6 protomers, and these 5 protomers, when it is present pentamer, it may be hexamer or it is 6, but actually capsomers are composed of protomers and capsid is composed of capsomers. So protomers are the lowest unit which join together to form a capsomer or capsomere. Sometimes in different books there are, and they said that, and then capsomers at last present you the helical, that icosahedron capsid icosahedron structure like this, okay. So remember this point: protomers, capsomers, and ultimately it will show you as capsid structure or icosahedron structure, geometric structure. Now coming to the capsid of complex symmetry. So complex means they do not contain either of these structure, but it may be a combination of both. So generally pox virus and large bacteriophages, mainly T2, T4, T6 even phages. This T even phages, even means 2, 4, 6 numbers represent T even phages. This contain a binary symmetry, means they contain both helical as well as icosahedral structure, as I've shown you in the earlier picture. In this binary symmetry, they contain the icosahedral head, within that the nucleic acid is present, and then a neck region, tail region, which is a helical part. So they need that structure for attachment and other purposes, right? So I hope you clear with the capsid structure. Now in the capsid part we have covered. Now coming to the envelope part. So envelope is very important section of the virus, but not all virus, because not all virus contains this envelope. Only enveloped virus contains this envelope. So why they contain it? So during entry or exit from the plant or animal cells or plant or animal viruses, they take up the lipid layer of the cell, that is the host cell, where they are entering or from where they are exiting. In that cases, that lipid bilayer which is present, some of the part form a membrane-like structure around the capsid structure. So it is called an envelope virus. Now this envelope contains several proteins, lipids and carbohydrates. And this particular structure is called spike proteins. Generally it is a protein structure, it may be a glycoprotein structure in composition, sometimes also called as peplomers. So these glycoprotein structures have several functions. Some work as an enzyme, some work as an what I can say, help for the virus to the cellular structure where they are infecting. So several functions are there for these spike and or peplomer structures. Now I will give you some special examples of spike protein that is present in the influenza virus, the neuraminidase and the hemagglutinin. These are two special spike proteins present in the influenza virus which help them to infect the or to get entrance into the host cell. After it, they can form the disease and show its pathogenicity. So one by one I will go. So this neuraminidase is an enzyme which breaks or cleaves the sialic acid present in the membrane, cell membrane of plant or animal cells. So it helps in exiting the exiting of the viral particles after the infection from a particular cell. So when they exit from the cell, they need to break the cell membrane, right? Otherwise they cannot exit it from it. So during the breakage, this enzyme helps the breakdown of sialic acid, which is present in the cell membrane as a glycoprotein structure, and then they pore formation, or they can leave the particular host cell where they are replicating. Coming to the another, that is hemagglutinin protein. Several tests are also designed on these spike proteins. As the name suggests, hemagglutinin. Hem means hemoglobin. Here agglutinin structure means the both virus and the hemoglobin attach themselves whenever infecting in the body, and they form a agglutination structure, hemagglutinin structure. So from there the name has come, hemagglutinin. So the red blood cells with the virions cause this hemagglutination phenomenon, and the spike protein from there named as hemagglutinine. So this is very important envelope enzymes present in the particular virus I have given you as an influenza virus example. Then the recent outbreak or the pandemic SARS-CoV-2 also contains spike proteins. Several diagnostic tests, molecular PCR, are designed on these spike genes. So this function of that spike gene is to get entry into the cell. Somewhere it helps in attachment, somewhere it degrades the cellular membrane for exiting or entering into the cell. So I hope this class helps you in your regular syllabus structure, either both for clinical and non-clinical students. So if you really think that helps you in your study, then please subscribe my channel for these highly detailed videos